To be here. Okay. My name is Gabor Marth from the Department of Human Genetics at the University of Utah, and you already know some of the peeps from my lab, Andrew and Al. Have you already done your some of your teaching, and he's coming back for the afternoon for the practical session. We've been involved in this course for quite a while now, and I'm going to be talking about variant discovery, variant identification, sort of like the state of the art. Starting out with sort of like the fundamentals, and then moving on to some of the current approaches and current ways we're thinking about trying to identify genetic variants accurately and sensitively. Okay. So starting out, genetic variations, as you know that Human Genome Project produced a reference genome sequence for humans, a series of 3 billion nucleotides, and this captures a very large fraction of the genetic makeup of every single human, right? So that's why we can talk about the Human Genome Project, because for the most part, your DNA is going to be very similar to what was sequenced back in 2001, right? Turns out that's it's over 99% accurate for any individual. Okay, so we can really call it as our, any one of us can call it as our genome sequence. However, there are differences. Less than 1%, but significant differences between any two individuals, or even within your own DNA. Can you harbor genetic variations, your own DNA? How? Can somebody tell me? Somatic mutations, one, and even inherited mutations. How many chromosomes do we usually carry in our cells? 2 chromosomes, one inherited from your mother and one from a father, right? And there is genetic variations between them. It's called heterozygosity. So we actually carry a large number, about 4 million genetic variants, even within the pair of chromosomes in our cells. Okay? So variant analysis is important for very many areas of science and medicine. One, population genomics, understanding how all these people are different from each other. Medical genomics, which the term in my usage means understanding what genes are involved, for example, in a particular disease, understanding the genetic causes of diseases. Personal genomics, where you turn around and ask, in this particular individual, what gene or genes might be involved in a particular disease or phenotype, as well as clinical genomics, which is similar, that really is restricted to understanding and diagnosing and treating diseases. Human genetic diversity is very substantial. So this slide comes out of the 1,000 Genomes projects, and what it tries to capture is that there is a significant diversity across human populations and even within human populations. Okay, with huge amounts of diversity being present in Africa, and some other populations are less diverse, depending on this is all determined by population genetic patterns and bottleneck patterns, etc. Why do we care about genetic variations? Well, first of all, they cause phenotypic differences, right? All these 4 people are very beautiful, but very different ways. And a lot of that has to do with their genetic heritage, as we talked about, heritable diseases. And it turns out that polymorphisms and genetic differences are characters that actually change very, very slowly. They turn over very, very slowly. So like the way the continents move. And because of that, it is possible to use the human polymorphisms to make inferences about demographic history. And there's been a lot of work about human migrations and the origins and peopling of continental populations, especially relatively recently, made possible by high-resolution genetic variation maps. Okay, so we talked about genetic variations, but what are they? So genetic variations come in different shapes and sizes. This is sort of like what many people think of first when they talk about genetic variants, right? These are single nucleotide polymorphisms, really substitutions from one nucleotide to another nucleotide, right? So some individuals with this chromosome will have an A here and others will have a C there, right? That's very simple. And these are, it turns out, the most abundant forms of, by far the most abundant form of human variation. But there's many others. There's insertion deletions where instead of maybe these C and T, there is nothing. You know, some individuals carry the C and T, some don't have those nucleotides. That would be a deletion or an insertion. And larger rearrangements, some of these examples like inversions here, these are inversions where a part of your chromosome, DNA in part of your chromosome flips and gets reinserted in an inverted orientation. That's a structural variant. Also, it's kind of hard to see in this slide, but translocations where a part of your chromosome, parts of two of your chromosomes get swapped and exchanged. Also, larger deletions where part of your DNA disappears or another part of your DNA gets inserted in multiple copies. These are repetitive elements. So there's many, many ways in which genomes can be different from each other, and these single nucleotide variants are only the simplest ones. Okay? So I actually going to talk a lot about this, and we're going to touch base a little bit about this. I'm just going to give a feel of how people go about finding these genetic variants between individuals. Okay. So how do we go about trying to find genetic variants? Well, by definition, if you looked at a single chromosome, could you, and that's all you had, would you be able to find variations? No, you need to compare things, right, in order to find differences between them. So variant discovery by nature requires comparative analysis of multiple chromosomes or multiple sets of DNA, multiple DNA sequences, representing, of course, the same region of the genome, so they are comparable. Because it turns out that human DNA is repetitive. I'm going to come to this a little bit later. So there's regions that look very similar, like maybe that this particular region is very similar to that. Differences between them are not genetic variations. Okay. Okay, so with this little introduction here, now I'm going to start talking about what's called reference-guided variant discovery, and that is how today most genetic variants, or to date, most genetic variants have been discovered. Okay? So the idea here is that for many organisms, and in particular the human organism, we now have what's called a reference genome sequence. And this reference genome sequence is not necessarily the genome of an individual, but it's a sequence that somehow represents a particular organism, for better or worse. The human reference genome is fairly comprehensive, and because it's been worked on for a long time, it's very important. So a lot of resources have been put into it. Some reference sequences from other organisms may be much less comprehensive, or much less accurate, primarily because of the resources that had gone into creating it. So the idea is that once you have a reference genome sequence, then you can use this as a template to organize, for example, sequences from two additional individuals. So if the yellow or greenish are DNA sequences from person A, and blue are sequences from person B, now you can localize these sequences relative to the organismal reference sequence, and with some caveats, you can be certain when you compare these sequences to these sequences, you will be looking at the same region of the genome in these two individuals. Okay, so that really is what the reference sequence gives you primarily. Okay? And then, once you have that, then you can go ahead and look in detail in these individuals and try to see how those sequences differ amongst these individuals. Okay? So that's really the fundamentals. So in terms of technical, how do you go about what's called read mapping? So first of all, why do we need read mapping? Because we can't read out entire chromosomes with current technologies, right? It's just there's no technology that can say, okay, I want a sequence of your chromosome 7, and then, you know, base 1 on chromosome 7 gets read out, and then you basically at the end, you know, just linearly read it out, and in the end you get to the last. So there's just no technologies like that. You have to read DNA sequences out in smaller chunks, and you guys, that was basically what this course is about. Okay, so that's why we really need to place those sequences now in the bigger picture relative to a particular human chromosome or the human genome. So in some ways, read mapping is very similar to doing a jigsaw puzzle, which I'm sure many of you have done, especially around Christmas time. It's a good time to do. The idea is that you have all these pieces, and then you have to put together a picture. Now, before there was a reference sequence, that's all you had, right? But with the reference sequence, they give you a box. So now you can try to take every individual piece and figure out where it is in the grand scheme of things. Which, of course, and this is why this is a good analogy, is an easy task for certain pieces like this one here with the tree. It's very easy to know where that comes from because that has such unique features. But others, like pieces of the sky, you know, that's where it's a little more difficult to place, right? Because they, you know, it looks like many other places in the genome. Okay? So it turns out that the reason for the sky, so what stands for the sky in the human genome, is repetitive elements in the human genome. And this is actually back from the original human genome, public human genome paper in 2001, reporting that over half the human genome consists of repetitive DNA. Some of these are small elements like signs or lines. Some of these are larger segmental duplications. Some of these are very complex duplications or multiplications, etc. But it turns out that it really is over half the genome that we have to deal with, and that's what makes up the sky and the grass in the previous picture. And of course, the problem is when you have a sequence that represents such a region, it's sometimes difficult to know where that piece comes from, right? Because it's similar to here and similar to there. It's a piece that is very difficult to place. Okay? So far, so good? So how do we deal with that? So Al and others have talked about various genomic data formats and talked about BEM files, right? Did you talk about mapping quality values? So the idea is, so the mapping quality value just expresses how confidently how sure you are that a particular piece that you placed here in fact comes from that place of the genome rather than some other place. Okay, that might be similar. The idea is, conceptually, that if you are able to somehow calculate a probability that you think, oh, it's 80% likely that this place comes from here versus 20% there and 1% there, then you have at least some probabilistic understanding, some confidence of your read placement. Okay? And so this is what the mapping quality values express. Not in probability space, it's basically, if you place a read, for example, here, the mapping quality value gives you the 10 base logarithm of the likelihood that that place was incorrectly placed. Okay? Similar to base quality values that give you a logarithmic likelihood that the base is not what you called it, etc. Okay? Now, from early on, this had been a big problem, especially with very short reads. Turns out that some of the technologies that we're using today, like the Illumina sequences, started, I think, maybe like 25 base pair. They were able to sequence 25 base pair fragments, and then like 36, and then the read lengths went up. So this was a huge problem, especially early on, and it still is a problem. But paired and read solves some of these. So the idea is, if you're sequencing instead of a single read, you're sequencing a longer fragment, then the chances are that at least one of the two ends will map into a unique sequence and can be confidently placed. And then you can choose between multiple possible locations for the other mate that maps into a non-unique sequence. So it makes sense? So this is how you're able to place read pairs better than you can place individual reads, and that's why you're seeing most of the sequencing today done as read pairs, especially in the Illumina technology. Okay? Longer technologies such as PacBio are different because the reads are already longer than what the two reads span, two Illumina reads would span, so the problem is much less acute. Okay? So you can place longer, say, PacBio reads much more accurately to the human genome. Okay, so turns out that there's a number of things that you have to deal with when we're talking about alignment. One is the idea of duplicate reads, and the reason for this is that most sequencing technologies, not all, employ PCR amplification during the sequencing process. So given a particular template of the DNA that was present in that individual's genome now has multiple copies, and you sequence a random assortment of those. So you might be sequencing multiple copies of an underlying DNA template. And so these are not independent observations of the original DNA. And if there are early PCR amplification errors, then these amplification errors get incorporated and then will look like genetic variations. Okay? So this is again a problem that has to a great degree gone away, and good sequencing now means that the rate of PCR duplicates is very low. But many situations you don't have enough DNA, so some very precious tumor sample, low purity tumor sample, etc. This is still a problem. You need to be able to identify those reads that of these 1, 2, 3, 4, 5, or 6, there's really only one independent observation of these of the underlying DNA template. Another problem, and I don't know if you guys had looked at alignments with IGV, you guys have looked at alignment at IGV, is that some regions look very beautiful and clean, and some others look sort of just messy. And that is because it's very difficult, actually. It turns out that it's in some regions, DNA complexity is such that it's very difficult to produce high quality alignments of the sequencing reads to the reference sequence. And in situations like that, there's a number of approaches with which you can try to clean up the alignments. What's the idea here is called local realignment. There's technical reasons for this. So when you align individual sequences to the genome, you're only looking at one read, a relationship between each read and the reference genome. You're not looking across the multiple reads to see how they relate to each other. These are not really true multiple alignments. So these local realignment methods try to employ some techniques that take into account what other reads alignments are to the genome, and take that information into account when creating cleanup alignment. Like, for example, in this case, there's really just a one base pair deletion here. And if enough reads say that, then you can try to see if the other reads are consistent with that hypothesis and realign that way, for example. Okay, so yeah.
Is this local realignment built into normal aligners?
To some degree. So every aligner will have some algorithmics of how to do the alignment, and some put some of these information in there already. Some are, there's programs that do nothing but realignment. Like, for example, the GATK has a realignment step where they take known insertion deletions, like for example, if this was a known variant, known insertion deletion, and they try to see if the reads map, would map better with that insertion. There's a number of different approaches for this, and each aligner will have some level of heuristics. The primary aligners have to be fast and performant, so they might not include steps that are computationally very expensive. So that would be a secondary process that you do if you need it.
So does this require you to spot that this might be a local misalignment, or is there like a file that gives all the locations of potential misalignments?
Well, I think again there's different approaches. How to detect it? There's basically counting misalignments, etc. in a region. There's ways to detect it automatically or by eye, and then certain types of aligners will deal with certain types of problems. Yeah, but there's not a one-time fits all solution to these, and it turns out that. We don't necessarily know whether the realigned version is actually better. It's sort of like a parsimony approach. If it creates something simpler, then you think that's probably truer. But ultimately you would have to do independent experimentation to know what the actual alignment is. And sometimes people do if the variant that is revealed is super important, for example, for a patient. So we're moving on now to the identification of SNPs, or single nucleotide polymorphisms. This is a very old alignment program that was actually used for the construction of the human genome reference sequence, but it's illustrative because it shows all the bases. That this is a particular situation where we're looking at a TG polymorphism or GT polymorphism. Okay. So the question is, how do we detect these given an alignment such as this? I basically just want to give you a cursory insight into how we think about when we try to detect genetic variants. Okay. So first of all, are we certain that these are genetic variants? Okay. What are the options here? Like if I look at, let me give you, so for example, if I look at this situation where I see a C and a T, or I see a G or a T, if I give you some more information, then you'll probably be able to tell me what you think. So in this case, in this particular scheme, capital letters mean very confident nucleotides, and the background, white background, the whiter the background, the more confident it is. Okay. So in this case, we have a very confident C against a very confident T. In this case, we have a low quality, not very accurately measured G versus a not very accurately measured T. Which one do you think is more likely to be a real polymorphism? Okay, without insulting your intelligence, it's obviously this example here. And in this case, it is more likely that either the G or the T is a sequencing error. Right, and that's why we're seeing this difference. So that's really the big, when it comes to small nucleotide changes, that's really the decision that you have to make. Are we, what we're seeing here, these differences that we're seeing, is that really true polymorphism, or is it just measurement errors? Is it sequencing errors? Okay. So turns out that when we observe an alignment like this, and look at this particular slice of the alignment where we see some apparent differences. So this is sort of like how I'm thinking, is that this is sort of like what we're seeing is that the top sequence, this whatever the true nucleotide is at this position, could be an A or a C or a G or a T, right? Those are the four nucleotides that we're working with. The second sequence, it could be an A, C, G, or T, etc., etc. So what we're reading out in the sequence is that the first one is an A, the second one is a C, the next one is a C, and an A, and a C. Okay? That's what we're seeing. That's what the data tells us. But it could be wrong, right? It could be that, for example, they're all wrong, right? They all misread the DNA. And in fact, all of them are T's, right? Is that very likely? It's not very likely, right? That would be like a lot of error. That would be a really, really bad base caller if that really was the case, right? So this is not a very likely situation, but it's possible, theoretically possible, right? What's also possible that instead of that these C's are all correct, and then this A and this A was misread, right? Would that be a little more likely? It would be a little more likely, right? Or it could be that, in fact, these A's are correct, and then these C's are misread. And because we see a low quality C, this, we could pretty much that we can imagine that. So that's sort of like this. That's sort of like how we're thinking, is that we have some observations. The base caller told us what it thought the bases were, but the base caller could be wrong. It tells us a base quality value that tells us how likely that it is wrong. And then we now have these probabilities to work with to calculate a conclusion. Okay? So, a priori, of course, what is more likely? For any given, if you compare two sequences, two individuals at any given position, is it more likely to be polymorphic, or is it more likely not to be polymorphic? Not to be polymorphic, and by what margin? So for that, and I know some of you know the answer, so we would need to know what the polymorphism rate is. So if I look at 1,000 bases in a human genome between two individual human chromosomes, how many variant sites would I see? One, about 1 in 1,000, right? So without knowing whether a site is polymorphic or not, a priori, I was a betting man, I would bet 1,000 or 999 against 1 that's not a polymorphic site, okay? So we need to have more evidence. We have to have enough evidence in the sequences to overcome that prior expectation, okay? So this, what you see here, is a mathematical formulation that I'm not going to go into detail about, but the idea is that what goes into evaluating this mathematically is the base cause of what we see and how confidently we see it. Okay, the more confident we're about the A's and the C's here, the more convinced we are that that's a true variation, and the less confident that these are really A's and C's, the less confident that it's the real variation, right? Polymorphism rate, so the prior rate. So if I told you that these are in fact PCR, these are sequences from a PCR amplicon, without expecting any differences between them except for PCR errors, then the expected polymorphism rate would be 0, right? So we would not accept any polymorphisms here. That would be an extreme example. If we looked in the region of, say, the human region that's hypervariable, for example, in the MHC region, we would expect a higher rate of polymorphisms, okay? And then also base composition, I don't want to talk about that, that's not important, but the depth of coverage. So if I see 1A versus 1C, I'm probably less confident than if I have 20 observations of the A allele and 20 observations of the C allele, right? So that how many observations we see also plays a huge role. So all that goes into a bag, well, a mathematical equation, and out comes a likelihood, and that's the likelihood that this, in fact, is a polymorphic site versus we're seeing these differences based on sequencing error, okay? So basically what you see in I think you guys learned about the VCF file, the variant call format file, that reports genetic variants after a computational extraction of sequence variations, right? So basically what makes it in there is a variant that has high enough likelihood to be reasonably confident that it's a genetic variation, okay? And often that likelihood, which is often termed as the variant quality, is also reported. Okay? So sometimes when you look at a VCF file, you'll see a variant quality, and that's what this represents. It's really basically just like any bioinformatics quality value, is the likelihood that it represents the log likelihood, the negative log likelihood, that that variant is not real. Okay? It's always the other way around. It's always the likelihood that you made an error with your inference. Okay. So I actually look back. So first, this framework we developed this years ago as part of the PolyBayes polymorphism discovery program. But I looked, and actually this is still up. This link is still up. If you click on that, and I want to show you, it's sort of fun. I just sort of rediscovered it yesterday. So if I can find my cursor. This guy here has a little demo, and the idea is that you can go and say, okay, assume that this is sort of a slice of the alignment, and then you say, okay, I want to see, suppose that I have 1A and, sorry, that was a C. I have another C, and then I have a T, and then another T, and they, base quality value 30, each of them base quality value 30. I can change that too, and then I get a answer, what is the likelihood down here. I'm 99.7% certain that this really is a true polymorphism rather than a, so it's done over 10 years ago, but for some strange reason it's still up and it still works. Okay, it's fun to play with. Okay, so it turns out that variant identification has come a long way since those early days, and with many new approaches. Like, for example, I'm just going to give you a couple here, if I can. So PolyBayes turned into multiple through other programs. Now it's called FreeBayes, and this is one of the premier variant callers today, developed by Eric Garrison, who was in my lab. And the idea is that we can now extend variant identification not to a single site, but everything that you can see within a read. So if you have 200 base pair reads, you can look for that entire region, variants in that entire region, and look for haplotypes in that particular region. So not only detecting individual variants like this, but you can look at their combination. So that's why FreeBayes is really a local haplotype caller rather than just a short variant caller. And the practical importance of this, of course, is, for example, identifying compound hets. Does anyone know what compound hets are? Correct. So the idea, the significance of that is that it may be that you inherited a deleterious allele from your father and one from your mother, but not at the same site, but at different sites. If you inherited in the same site, you're homozygous for that variant, and then you, for example, have a recessive disease, right, where you have to have 2 bad copies to get the disease. Sometimes you get at one site and you say, oh, this person has the deleterious allele. You get another site, it's, oh, this person has the same person has another deleterious allele. But if they're on the same chromosome, they're knocking out, this one already knocked out gene function, that one is already beaten at that horse, there's still a good copy. Right? Whereas if they knock out different copies, then there is not one good copy of the gene. So that's why it's very important to be able to decide whether 2 variants or the phase between 2 variants, whether they are compound heterozygotes, that is, they're knocking out different chromosomes, or, as in this case, they're on the same chromosome. And not only that, in this particular case, the overall effect of these 2 deletions is in fact a frame restoring situation. So this is the in-frame deletion of 6 amino acids after which the frame is restored. So you would individually conclude that this is a frame-shifting deletion, that's a frame-shifting deletion if they are on different chromosomes, but put them on the same chromosome and now become something that the expected impact of it is probably not as deleterious. Okay? So these are the type of things that you want to know. And of course, you can only do that within read length. If these are, the first exon of a 100 kb gene and the last exon of a 100 kb gene, then you don't really have that type of information in the reads because they don't span all those regions. Okay. Okay. So what? Okay. So then, now this slide. So. All this research is very collaborative. We basically work with all our competitors and all that, and to move the field forward. So GATK is the other very important variant caller. And you guys, are they going to use GATK in the practical sessions? No, but you could, and that's the premier variant caller. So I just put it in this slide to give you guys an idea of what the accuracies are for SNP identification today. And this comes out of a recently published paper with Ira Hall and Aaron Quinlan, who talked yesterday, and he's going to be teaching more in this course. The idea is that for single nucleotide polymorphisms, your sensitivity is close to 99%, 98, 99%. Okay, so out of every 1,000 SNPs, you can pick out 980 or so. Okay, so it's very high sensitivity, and the specificity is also very good. So basically, well over 95 or 99%. So out of every 1,000 variants that you call, only a handful may be wrong calls. Okay, so SNP detection has come a long way, and it's very, very accurate now. Okay, it turns out that insertion deletions are not at the same level yet. We're looking at basically a sensitivity value of between 85 and 90%, and actually the false discovery rate is about 1 or 2%. So it's not quite as accurate as for single nucleotide polymorphisms. Okay? Yeah.
Freebase is better than GATK?
Well, it depends. So it's all, if you look, for example, you'll see that the sensitivity is better for freebase with a given set of parameters, but the false discovery rate is worse. So basically, it's all a compromise. So where is it that you're going to cut it? Yes, if you want something more sensitive, you might use one program or one set of parameters. They're very comparable, actually. You can set parameters that will give you similar compromises between sensitivity and specificity. Yeah. Any questions about SNP calling, etc.?
Is this data across the genome or the exome?
This one was genome. And it's against the genome in a bottle, which has a bunch of caveats. Those tend to be the variants that are fairly easy to call anyway. But basically, you have to compare to some sort of truth set. I'm going to talk a little bit about a program that our own Andrew developed that can detect SNPs anywhere. Of course, it's very difficult to then prove that they are real because they're in the most impossible parts of the genome. Okay, so then we move on to detecting structural variations. So structural variations are what? So I'm just going to throw this picture up again. So these are larger events that basically move around larger bits of DNA, like flipping of a lot of like a megabase, or translocation, or duplication of a region. Instead of 1 copy, this region now comes in 2 copies, and instead of 5 copies, it now comes in 6 or 7 copies, etc. Okay. So it turns out that whereas short variants, you detect short variants in the base-wise alignments. So what you do is you look at all the reads, you align them to the genome in this case, and then you look across the sequences. and you say, where like this C in this sequence corresponds to that C in that sequence, and then this G corresponds to that A, and now I detect differences between them. So basically use the reads and what you read out in the reads to detect variance. When you have larger variance, this becomes almost impossible to do. Okay? Just because, and this is something that I'm going to come back a couple more times in this talk. But the idea is that you, for example, have a very big deletion, for example, like this. This is the reference sequence that has this segment of the DNA that's missing from the individual you're sequencing now. Then the signature that you get is just there's no reads aligning to that region from that sample, right? So it's not that you have reads to detect in that variant because there is nothing aligned there. So you have to use that fact itself. Okay, so that's sort of like the easiest to understand signature of a genetic variant. Also, if you, for example, in the same situation, if you now have read pairs that, we say the expected fragment length is maybe 500 bases, but then in this sample, this segment is missing. When you take these two end mates from that fragment, they'll map on two sides of that deletion. If it's a 1 kb deletion or a 2 kb deletion, it would look like the fragment length between these two end mates is like 2.5 kb or 3 kb. So what's called aberrant fragment length or apparent fragment length and read pair distance is another signature of some of these structural variants. Okay. Split reads. So split reads is a situation where, for example, in this case, you have this segment in the reference sequence is not present in your sample, and of course the read will read the sample's own DNA. When you align it back, it would look like it aligns here, and then it stops aligning, and then it starts aligning someplace else, so sort of skips a region. So if you have software that can do what's called split read mapping, then you can detect this read is indicative of that sample not containing this particular region in its genome or his or her genome. Okay? And then another way that's gaining, where methods are really getting developed, good methods are getting developed, is de novo assembly, where the idea is that you try to assemble the genome of this sample, and you have the complete sequence, then you can do basically comparative genomics, just compare it to the reference sequence directly. Okay, so you're not using the individual reads, but you use the assembly created from these reads to try to detect genetic variations. There's all kinds of caveats with that. It's not that easy to assemble an entire human genome. What is more commonly and more successfully done is assemble reads from a particular region, including on-map reads and on-map mates, etc., and try to reconstruct the sequence of this individual in this region, and then compare that to the reference or some other individual. Okay? And it turns out that there's another method that I'm going to be talking about later, and that's also completely reference method, and that's the method that Andrew developed. So now to give you an idea, just give you a little taste of how structural variants are discovered, we're going to talk about a project that I'm very familiar with because it came out of my lab, and that is detecting what's called mobile elements. So what are mobile elements? Mobile elements are sequences that are highly repetitive because they are elements that either can duplicate themselves or can borrow the host cell's machinery to duplicate themselves, and they can insert themselves into a given genome in many, many copies. That's why they're called mobile elements. It turns out that some elements are in fact still active, which means that certain ALU subclasses, for example, you still see that, say, every 25 or so live births, you'll see a new element inserted in a child's genome that was not present before in anybody's genome. Okay? So these elements are still active, and because they've been active recently, many of these elements are polymorphic. The idea is that what is polymorphic is that some individuals have it, some individuals don't have it. So once a new insertion occurs, then that child has progeny, etc. So now that character, that allele, is now in the population, and it can either die out, like many alleles, or it can be fixed, that means that now everybody has it, or it can be polymorphic in an in-between stage where some individuals have this allele and some individuals have other alleles, right? So these are polymorphic elements. And it turns out that if you compare two genomes or two chromosomes, there's about 2,000 events that we can detect. Maybe now we can detect maybe a little more now. And when we started this study, we didn't quite know how these elements, what were the population genetic properties of these elements? Do they segregate like SNPs or some other way? So the way to, the funny thing is that you could actually detect some of these mobile elements, insertions, in a sort of like in a reverse type of way, because the human reference sequence, in fact, includes some polymorphic insertion alleles. And when you look at additional samples that you sequenced that don't have that allele, then it would appear these elements that are insertions in the reference appear as if they were deletions in the sequence in samples that you sequenced. So you can really detect in a reverse way deletions, mobile element deletions in a sample that you sequenced are in fact insertions in the reference. And why do we know this? It's because there's no known mechanisms to precisely excise these elements. So once the element is in, it's in. It's not like SNPs, which can be deleted or added, or nucleotides. These guys, once they're in, they're in. You can still delete them if you delete the whole region, but typically they don't get deleted. Okay? But there was no ways to find situations when this allo element is not part of the reference, but it's part of the individual that you're sequencing. So the two methods that we used was so think of a situation where you do paired-end sequencing of the individual in which you want to discover these insertion events. And the paired-end sequencing will normally just, the two pairs will align to unique sequence. But in some situations, you'll see that one end of the sequence actually is in the inserted allele. Okay? And then another one will map to the genome sequence as we know it. So this is actually somewhat, it's not a trivial situation because you see a lot of the elements like this in the genome that are not polymorphic and not insertions in that sample, right? So you see, because the reference sequence already contains a ton of allos. So you really, it has to be done with care to know that this particular element here is really not present in the reference sequence. And that's why it's unmapped, but it does map to the known sequence of the insertion element. Okay? Another way to do it is to look for reads that align really well to the reference sequence, and at some point they stop aligning to the reference sequence but start aligning to the sequence of these insertion elements. So this is a split read approach. That's a read pair approach to detect the same type of events. Okay? And it turns out that there's a lot of these. If you use these as these mobile element insertions as evolutionary characters, if you will, they segregate just like SNPs. Okay? And their site frequency spectrum is pretty much the same as for SNPs. Yeah, you got a question.
These racial populations, or?
These were European populations. So these were the 4,000 genomes population groups that were first used. So that's CEU was a European derived, YRI is from Yorubans in Africa. CHB is a Han Chinese population, and from Beijing, and this is a Japanese population. Yeah. And you can see that the Japanese and the Chinese cluster close together. The Yorubans are pretty far, and the Europeans are in a different group based on this principal component analysis. Again, not rocket science, it's just, but what's really interesting is that these guys act just like SNPs. They come into being, then the likelihood that they get fixed in a population is very low, but some do get fixed and some become polymorphic, and their frequency in the population follows the theory, very similar to SNPs. So in fact, here we could compare them. The mobile elements are the continuous lines, and the whatever, I forget, I'm drawing a blank. The non-continuous lines are the SNPs, and the side frequency spectrum is identical. It's super nice. Okay, so this just gives you an example of how you can go about not using base alignment at all, but just these signatures in how you map the reads or pairs of reads to detect these elements. Okay, then you can go back and validate them, like you can design primers that read across the boundaries, and then you can experimentally validate the what's called the breakpoint sequences, the event boundary sequences, and you can be certain that those elements really are where you predicted them to be. Okay. Okay, so now I want to move on to a different topic, somatic mutation detection in tumors, but I want not necessarily because, but with two purposes. One is just to give you a little idea of how somatic mutation detection is different. But this is sort of like a way I want to introduce some of the new technologies that are being developed for variant identification. So cancer is largely a genetic disease, right? But not in the way often people think about genetic diseases. They're not necessarily inherited. You don't necessarily inherit a causative allele or a breast cancer mutation or something that predisposes you, but you may acquire it. Okay, so these are somatically acquired, often somatically acquired mutations, like for example, if you smoke, or cosmic radiation, or some other environmental exposure, or just random chance mutation. Okay, these are somatically acquired. So, but just because a mutation is somatic, it's still genetic, it still affects your genome. Okay, so many of the methods that we use for studying inherited variation still apply. And so tumors, what they do as a result is they are able to evolve rapidly, which means, for example, that once you develop ovarian cancer, cancer is able to evade chemotherapy and develop, for example, chemoresistance. Or a tumor may change to the degree that it loses its sense of location and loyalty to an organ and can establish metastatic lesions, distal metastases in other parts of the body. Okay? So all these come from the fact that the tumor genomes can evolve to great degree. So genetically speaking, if you look at the germline variants, each cell in a human body will carry a complement of mutations that you inherited from your parents. Like you may have inherited the triangle allele from your dad, and and then now every cell will have that allele. Or you may be homozygous for this circle variant. You inherited a copy from your mother and your father, and now every cell in your body will have those variants, right? On the other hand, for somatically acquired variants, a cell in your body may acquire a particular variant, and then that cell then gives rise to, other cells, etc. That progenitor cell gives rise to other cells, and new mutations come into being. And so ultimately, instead of having a very regular pattern of mutations, that is, you either don't have a variant or you have that variant in one copy, in which case the overall prevalence of that allele in your cells will be 50%, or you have two copies and it will be 100% in all your cells. Whereas in tumor, some mutation that somehow came in early and it's now present in many cells, or maybe at higher frequency, and there might be some very rare variants that are present in your tumor. Okay? So you don't have this regularity here. It turns out that somatic mutations are, it's an aside, is they're not restricted to tumor cells, right? Every cell in our body has somatic mutations, but they're not as important until they do something that isn't undesirable, right? Like become cancerous, etc. And there is also some conditions that are characterized by what's called somatic mosaicism, where a particular somatic mutation may be at high frequency, like, for example, port wine stain. It's like brown stain on your skin. I think the underlying cause is somatic mosaicism, that some of your cells have a particular somatic mutation that causes overpigmentation. Okay? But it's not necessarily a cancer. But it is very important in cancer. So the idea here is that in your soma, in somatic cells, every cell has the same variants, whereas in tumor cells, there's divergent and evolving groups of these cells, or what's termed as subclones. So what's a subclone is basically is a group of cells that has the same complement of somatic mutations. Okay, and so this is one way to think of tumor evolution, of how these subclones then acquire new mutations and maybe get a selective advantage, and then that subclone may acquire high frequency, maybe completely take over the tumor mass, etc. Okay. But I'm not going to talk about subclone reconstruction, which of course is a favorite topic of mine, but I just wanted to use this example to talk about variant identification. So the problem with detecting some of these tumor variants or somatic variants is that some of these are very low frequency. It may only be, say, 3% of the cells has a particular variant. Okay? Now, if you only 3% of the cells have the variant and your sequencing reads have 3% error rate, now you're at a place where the signal, which is genetic variants, somatic variants that occur in a small fraction of the reads, are now comparable frequency to the noise, that is the sequencing error rate. So that's very difficult to detect. Okay? Also, another sort of like more technical aspect is that if you think, so when we characterize how successful your variant caller is, for example, we look for things like false discovery rate or false positive rate, etc. Now, it turns out that when you're looking at inherited variants, as we established, the inherited the variant rate is about 1 in 1,000 nucleotides. Okay, so there's maybe 3 million or 4 million or so in a genome. Somatic variants are much rarer, so the number of how many somatic variants you have in a cancer is hugely dependent on cancer type, and also what types of variants. Some variants, it's only all point mutations, very few structural variants. Some others have a rampant structural variant. But 10,000-15,000 somatic variants is about the ballpark, or 300 per exome. But the idea is that if you make 15,000 errors, then 15,000, compared to 15,000 true variants, that's 100% error, right, or a 50% error rate. But 15,000 errors versus 3 million variants is a minute error rate. So you can basically use the same exact calling process, and in a context where you have a lot of variants to call, and you call most of them right, you're gonna be a hero. But if you use the same software where there's much less signal to detect, you're going to be a villain, right? So your program will not work very well. So this is an important sort of consideration. So you have to be very accurate for tumor variant identification to have false discovery that's acceptable. Okay, more so than for detecting inherited variants. Okay, so specifically, I'm going to be talking about a situation where that we're recently working on with Aaron and others in the lab, in my lab. The idea here is that we had this patient, this is a refractory it's a metastatic breast cancer patient. The idea is that it's sort of fairly late stage. The patient is given some medication, the patient then responds, tumor load goes down, then the tumor load comes back, then get another course of chemotherapy, tumor responds, etc. And the idea is to try to prolong it as long as possible. Okay? And ultimately, often the patient dies after a few courses of this. So our collaborators. So in this particular case, so this is an example that sort of like meshes with what I just said about 15,000 point variance in this individual, about 300-400 in the exome, and about 80 missense variance in a patient like this. So this is sort of like the lay of the land. So the first point I try to make is how we can go about try to improve the sensitivity of detecting these variants. Yeah.
This slide and the previous slide, you said that there were about 15,000 somatic mutations in the genome of cancer.
Yeah. In this. Yeah.
In exome.
Oh, sorry. That's wrong. No, this is wrong. These are.
My question is
yeah, no, that's
the
right.
mutation rate higher than, because I mean, you think that in exons or in the exome would be 1% of what's in the genome. That seems.
Yeah, how was that? What are these numbers?
In the exons than in the genome?
Well, in general, in general, you see about per base about half the number of variants in exomes. Then you see, per base elsewhere in the genome. So what's 1% of 15,000?
150.
Yeah, no, that's fine. That's fine. Yeah, that's what it is. It's about 2%. Yeah, this is about right. Yeah, the exome takes about 2% of the genome, so we have about roughly 2%.
You're not calling the whole, non-coding genome because you masked out lots of repetitive areas.
Yeah, you're just calling the coding, the coding parts of the gene, not everything, from the gene beginning to the gene genomically.
Yeah. I thought it was close to, the exome was closer to 1% of the genome than 2%.
I think it's more 3%. That's my understanding. Yeah. Yeah, so these numbers do add up. And about 80% of them are missense or nonsense. So,
I have a question.
yeah.
So on the last slide, you showed 5 different samples from the same patient.
Yeah.
Did the mutation rate accelerate as the cancer progressed?
I'll show you. So it's okay. I'll skip here. Okay, hold on. I'll skip here, here, and then go back. So this is sort of like how it was. So this one here is the normal sample, and then these are 4. Actually, the whole genome sequencing we did 4 time points, and you can see that in the relative to normal, there were in the primary tumor there were 10,500 variants. And then a relatively small number of additions. So between this B1 first time point, second tumor time point, there were 621, and another 1,000, another 43. Okay, so most of the variants were present in the primary, and then a small number then got added later. Is that? Of course, whether it's biology or it's just timing, it turns out that before you had a primary, you had a lot of time to evolve those somatic variants, but then you were sort of like on the clock, and then we got sampled every few months, so there's less time to have new variants. That's sort of like Tom Petty, you know, like it's easy to make your first album because you had your whole life to make it, but then you're expected to put out an album every year, so that's kind of difficult to. So tumor thinks the same way. So, all right, but going back, so here the idea is, though, is that suppose that we find a variant at this particular time point at fairly high frequency, 30% or something like that, and maybe, you know, we can ask the question, and we didn't see it before. So biologically, it's very important to know whether it really came since the previous sampling, and it was not present at all, or was it present but just very low frequency and now it got selected for. Okay? Also, you can think of it if you applied some chemotherapy and you might ask, okay, maybe I don't see this variant here, assume that this is not here, but has it really disappeared or has it just become very low frequency? Have I been able to really eradicate it or just took it down to a lower frequency? Make sense? So was this present here? Was this present there? I could put a question mark there too. So the technology, as it were, to help with that is what's called joint calling. So what's joint calling? The idea is that instead of looking at just a single time point and calling the variance, I borrow information across different time points. So so this is sort of like the joint calling in populations is that if I look at these individual variants in this individual, that individual, etc., and I see them only once, I will be less likely to believe it than if a variant recurs in more than one individual, right? So of these ones, this variant is the one that I tend to believe most likely, okay? Because I see it in more than one cases, okay? So similarly for tumor variant, the way you apply that is that suppose that at this time point, we see this variant at high frequency, and our previous analysis wasn't convincing enough to call these when we looked at them in separation, but now we look at them as a unit all together in a single variant calling step. Because I see this variant here, now the likelihood that what I saw here being a signal rather than sequencing error is higher, so I'm now able to say that, no, this variant was already present in this time point and hasn't completely gone away after the next course of therapy. Okay? So in separation, it would be difficult just because, signal to noise is not going to go away. But this is a way to sort of bootstrap calling at this position from information gained at another time point. Okay, so this is one way to, yeah.
Do any other callers use this joint calling method?
So there's some joint callers that not for tumor. So tumor calling tends to be you do normal versus a tumor sample, normal versus another tumor sample, normal versus another tumor. Even some of the state of the art, like Mutect, is the state of the art in variant calling. Variant calling is pairwise. So, but this really gets you extra power. And I can show you that, for example, this is a situation where this is a 5-base pair insertion, and what ends up happening is that pairwise calling detected this variant here. So basically what you see here is how many reads report the reference allele versus the mutant allele, the alternate allele here. And then with pairwise calling, the first time you convinced that it's here, it's at this time point, whereas really it's present earlier. So if you had this information that you know that this allele is here in this time point, you would have been confident calling that particular allele at an earlier time point. Okay? And this is sort of like the statistics. So the idea here is that if you ask out of all the time points that we had, in this case 4 time points, how often you find a variant at another time point that was on-called by pairwise methods, you'll see that a large number of cases, we found a large number of cases where you can detect that variant at a different time point, not only where you could call it in just a single tumor sample, okay, when you looked at each tumor sample in separation. Okay, so that's number one. So that's lesson number one, joint calling. Always try to call your samples together because you have depending on the biological concept, you can borrow information across different samples. It turns out that for population calling now, that's for 1,000 Genomes Project, we called all 2,500 samples together. But for tumor, this is not the state of the art yet. So, okay. So then the next one, next technology I want to talk to you guys about is called graph-based variant detection. So many of you guys might have heard of the BWA MEM, might have heard of when you talk about the genome sequence, there is the newest genome version, GRC38, has these alternative contigs and alternative assemblies. How many of you guys heard of that? Couple of you. Okay. So the idea here, and I'm going to go back to the previous, my starting analogy here is that suppose that so reference-guided methods have limitations for diverged variants because if you look at two individuals and they look like this, and maybe the differences might be little specks that you on the box, some little printing errors between the two boxes. It's easy to do reference-guided because even if you had a little speck, you would still be able to place that piece where it is. But if you have a situation like that, now the piece that represents this tree does not look like that, and vice versa. Okay? So one idea is that if you know that at this some individuals, instead of this tree, you see that tree on the meadow. In other words, if you know that some individuals will have a particular sequence and other individuals will have another sequence, you can augment the reference sequence, and instead of having just one box, you can have sort of like a box where you have the box, but you cut out the tree, and then you say, oh, it's either this or that at that particular location. Okay, so this is sort of like a graph that is the same everywhere where there's no differences between individuals, and then where there is substantial differences, you basically you provide a representation of both alleles. Okay, so I brought up GRC38 because the new reference actually attempts to do this. They provide you with sequences that represent these conifers, that is, and other sequences that replace, the larch or whatever this is in some parts of the genome. Okay, but this can be taken to an extreme, really. And it's possible, so our version of this is what we call the variant graph. The idea is that if you know what sequences are in a patient's tumor at a given time point, you can construct a graph, and then you can go back and try to fish out read evidence for those alleles at other previous time points. Okay, so the way you would work that, so again, just to see how this works, suppose that this is an allele that you found in time point 3, but you didn't call it at time point 2, you could say, okay, now I'm going to go back to all the reads from time point 2 and map those reads back to this augmented reference sequence now that has the new allele. And then every time I see something like this, oh, this is the reference, this is no new evidence. But every time I see a read that maps like that, now then I may be able to fish out additional reads that were not originally mapped because they were so different from the reference, but now that I provided the right template for it, now I'm able to capture these reads. Okay, so that's the idea, so this is what we do, and with this, in this particular, this technology that can be used in multiple different ways. But the idea is that in this case, what we want to do, we want to fish out reads to know whether a particular allele was present at another time point. Also, more accurately estimate the allele frequency of that variant in the tumor mass, because it turns out that's important for understanding the subclone structure in the tumor. So it's important to know what the actual cellular frequency of a particular mutation is in, say, the primary and a relapse sample. So we need to have this value accurately estimated. So this is a situation where we have, and I'm going to focus on this particular area here, is that the traditional mapping revealed 14 mutant alleles that had the somatic mutation and 48 reference alleles. When we went back with this graph method and offered the alternate allele as a branch of the graph and mapped all the reads back to it, we fished out an additional 17 reads. So this means that now the frequency of that allele became double from what it was before. Okay, so this is a significant change. So now that variant will be clustered somewhere else, and it now belongs to a different subclone than what we thought originally it was. Okay.
How is it that your reference allele counts are also increasing?
Well, that's an interesting one. So that's because the original mapper used a specific way of when it aligns a particular read. And we did a different one for the remapping. One that is a little more straightforward and works in a directed manner. Yes. So that's actually an interesting observation that we were surprised to see, but we increasing the reference counts in some situations as well. Yeah. And this is sort of like a dot plot. So you can see that in many cases, there were no differences, but there are many alleles, many mutation sites where the allele frequency from the original call gets doubled or tripled. And if that happens to be an important mutation, you want to know that. Okay, and you can see there's definitely a shift to higher allele frequency of the somatic allele with this method, because you're really able to fish out all the reads that were important there. Okay. So then, now take this to the next level. So using the graph, it still requires that you know what that allele is so that you can put it in the graph. But it turns out that some variants are just very difficult to detect, and so we have to deal with that too. So turns out that very specifically, if you look at detection efficiency and where current software is, so this is sort of like the range of single nucleotide variants is 1 base pair. Short indels, what I call short indels, are maybe up to 5, 6, 7, 8, 10 base pairs or 15 base pairs. And then medium indels are anywhere between like 20 to 200 base pairs. And then we come into the structural variant domain, like alu insertions, 300 base pair, deletions, 500 base pair and longer, etc., until we come into like these long copy number regions, maybe megabase copy number deletion or amplification, whatnot. And it turns out that that short variant callers are very good up to about 10 base pair, and then their performance degrades just because you now have to detect these larger deletion events in mapped reads, and it's just getting harder and harder to map those reads there and align them properly. But the structural variant callers don't kick in with high efficiency until about 200-300 base pairs. So there's this blind spot here for events that are between 40 and I just say a different number, but approximately 40 to 200 base pair events. So this is actually a very difficult area there. It's very difficult to detect variants there because the patterns are not specific enough yet that the structural variant callers use, and the alignment-based methods are losing power. So another way to detect variance, instead of suppose in this particular situation, instead of trying to take the fragments that represent each of these and map them back to the appropriate box, is just to compare these two directly without an intermediate step of mapping to a reference, and try to somehow just focus on regions that are different between two individuals. And you can imagine in tumor sequencing it's a well-suited approach, because what we want to know in tumor is what are the somatic variants. We don't care about inherited variants in the first approximation. We just want to know what's different between the tumor tissue and the normal tissue from that same individual. Okay, so a method called Rufus is a reference-free variant detection method that does this, and I'm going to go through this. This is your TA, Andrew, here, developed this method. Just go through it very quickly. So the idea is that what we want to do is two individuals, Mr. Blue and Mr. Red, and or two tissues. Maybe this is normal tissue, tumor tissue, and what we just want to focus on those regions in the genome in these two genomes that are different, and don't care about anything that's very similar. Okay, so the idea is that we want to reduce all this data down to a very, very small subset of the data that is now represents differences, and then want to compare those and get the mutations that way. So the way it works is that you start out with this cloud of reads, so the whole genome's worth of reads. Have you already talked about this? Okay. Were you going to talk about this? Okay. All right. All right. Good. So we do clouds of reads, and then we create what's called hash tables. So the idea here is that we chop these the reads from the blue sample and the red sample into, say, 25 base pair fragments or hashes, and count how many times they're present in this sample and that sample. Okay, so maybe this AGT, CG, the first one, is present 46 times in this sample, and maybe actually, huh?
46 in one sample and 46 in the other sample.
Yeah. But then this one is 54 times in this sample and 58 times in that sample. Okay, so some different number of amounts. And then the way you identify true variance, you look for situations where one particular k-mer, this one here, is present 22 times in one and 0 times in the other, right? Or very small number of times in the other. So this is DNA that's unique to one sample and it's not present in the other sample. These ones here before were present in approximately the same representation in both of the samples, so they're really shared between these two samples. So this allows you to zero in on things that are unique to one sample or the other. It turns out that what about sequencing error? Well, in the case of sequencing error, you have situations where both have very low counts, just because that's a sequencing error. It's not really present in any of the samples. Okay, this was just the machine made this read, made this sequence. It's not really present in any of them. And then unless the errors occur systematically, you'll only see a very small one, it will be a one-off, rather than if you systematically, the machine makes that same error over and over again, then you of course get fooled here. But the idea is that in these situations, you throw that data out as sequencing error.
You don't get fooled if it's systematic, because then it's in both samples.
Uh-huh. Right. That's true. Yeah. And then basically then you have this way you're able to eliminate most of your data, you end up with a fraction of a % of your reads. that you started out with, you assemble those, and then you detect. Basically, now you do comparative genomics to compare the two samples together. Okay, so there is a unique section of the DNA on the tumor sample. Now you can, for example, map it back to the genome and say, okay, where is this approximately, and then you can characterize that variant. So this is actually has been very useful, especially in this tumor sequencing project that we're doing. This is a particular 77 base pair deletion, so falls right in that crack, right between, the medium range indels. And you can see this one here is the normal, and these are all the tumors. And you can see that this, Andrew. Oh, no, sorry, that's B0. Sorry, things are shifted here. So this is B1. So you can see that it's present in all the tumor samples, but not in the normal. But this was not detected by any other method. So you have a lot of these situations. Another one here, an 807 base pair deletion, etc. And it turns out that what we did here is we looked at short variant calling, and just worry about this side of the figure here. These are single nucleotide variants. These are short deletions. You can see that short variant caller, that is FreeBayes, is able to actually detect most of those variants and very, very high sensitivity until it peters out between 21 to 50 base pairs. And then the structural variant callers, this specific case, Lumpy, which is written by Aaron Quinlan's group, sort of picks up and is very, has good sensitivity, after about 200 base pairs. But there is this little swamp here, and then this, so reference-free detection can really fill in the gaps there and detect these medium range variants at high sensitivity. Okay. All right, and with that, to my lab and take any questions. Yeah.
So with Rufus, are you still throwing out duplicate reads before, because I guess, so you're just taking pieces of the reads and comparing them to each other?
Yeah. Yeah, and then yeah.
So you still throw out the duplicates first, so you don't have a preferential.
Andrew, why don't you?
So there's no mapping or anything, so you don't know what the same is not a big.
No, but so one thing is that based on the k-mers, you actually go back to the reads. So you identify which reads contributed the k-mers, and that's what you use for assembly rather than the k-mers themselves. But duplicate for exome sequencing, duplicate is a duplicate.
Duplicate is a little bit wrong. The issue is you're removing everything that's the same. So it's kind of the opposite of sequencing, where you have to match things to the reference first, and then you're going to do them. With this one, you want to find things that don't match.
Right.
So it kind of has the opposite problem where duplicates, since they're frequent and occur everywhere, then they just get thrown out anyway. Sort of, it's focusing on the other end of the spectrum of reads to find the reads that are unique and that don't match anything else.
So then you not produce BAM files, you just take a fast view and put it directly into these programs that compare them?
So it's
Yeah.
not fast view. You can start with BAM, but it's still basically fast views.
But then there's a few technical things, like you detect these variants, and some of these are totally unmappable regions. So you know that this patient, this sample has something that's not in the other ones, but you have zero idea where in the genome it is because you just can't map it, right? It's a contig that is not part of the assembly. So that's one problem. But then you want to somehow report it. Where is it? So you know where it is because you are able to take the sequence that you created that's unique in, say, the tumor patient. You can map it back to the genome reference sequence so you know where it is, right? So then you can report it in a VCF format. So it does include a mapping step, but this doesn't drive the detection. It's sort of like just a reporting localization step. And then you also you don't get allele frequency as well either. So then we use the variant graph then to say, okay, now let's take that new allele now, map reads to the graph, and then now we can measure the allele frequency. So this is the engine of detection, but then there is other steps that you have to do to sort of like embellish and add the additional information that is required to characterize a variant. Yeah.
Have you tried using these, like Rufus, I guess, a reference-free method from genome sequencing from non-tumor samples from the same individual?
Non-tumor say this again. So
I'm just interested. 2 skin
oh,
biopsies
yeah.
from the same individual.
Right.
You talked a little about. We're not talking about cancer, not tumor normal, but just 2 samples
right.
to look for, to see if they can detect differences in 2 samples. Just highly sensitive.
Yeah.
Low frequency variants
Yeah.
that are just in normal tissue from the same individual.
So low frequency is not the sweet spot necessarily, because you still have the same signal to noise issue. But rare is the sweet spot. So we didn't do that, but we did do, for example, trio sequencing when you have parents versus child to very sensitively pick out somatic variants. But we did not have
in that case,
actual.
you're using you mean de novo variants?
De novo variants, de novo variants that are not in the parents but are in the child. Now the thing is that
Can you do that by actually, like you're saying, sort of mapping not to the reference, but mapping against the parents?
Yes.
And showing a difference for the parents.
Exactly. Yeah. And there's not this intermediate that you have to map to the reference first and then compare, but you direct compare. Yeah. And it's super sensitive for those somatic variants, the de novo variants. Of course, it finds things that are not mappable and are difficult to prove that they're real because they're not, you can't map them to the current reference. Yeah. Yeah. Yeah.
So then you could take your output just from Rufus and say, another 5 years down the road, a new version of the genome comes out and it has some of these regions
Yeah.
that are mapped. Then you can just take what you got when you ran Rufus originally, because you didn't
Yeah.
you didn't use any genome assembly to do that.
Yep. That's the idea.
Do you find things mapping to triplet repeat regions like Fragile X or can you find that?
I mean, we don't.
Yeah.
It's one of those things like, so if you have largely duplicated genes, there's a variant in one of them.
Well,
We
sometimes
don't know
that's
which one it is.
Yeah, but sometimes if you just know that there's a difference in that
Yeah,
stretch, then you
yeah.
can go back and figure out exactly what.
Yeah. And fusions, you detect. I mean, what you will detect is really what's truly different. So if there's a large structural variant, like something, like an inversion, then we have markers here. So if you have an inversion, for example, so here's your genome now. It looks like I don't know how to, like it's like flipped relative to this. Then what, then the mid part is no different. The only thing that's different is around the insertion sites, what's called the event boundaries or breakpoints. So it will basically create a contig around here and around there that's unique to this sample, and then you can map it back, and that's how you characterize. Oh yeah, that's right. That's an inversion. It really just detects what's different first, and then you have to characterize it afterwards.
And the thing is, it's relatively not sensitive to the size of it,
Yeah.
so you have to map across it, just as you map the
Yeah.
So why is this ability to call SNPs here?
Sensitivity of SM
Yeah, calling the SNPs
with
with this
Oh yeah, so that's a good question. So you're asking, well, so it's not, so basically what you're finding is that there is this fraction of the variance that Rufus doesn't call, but FreeBayes does call. So some of those are artifacts of the program, and do you want to explain?
So some of them are artifacts of PolyBayes that always will confirm correctly because we're doing everything in reference space. The one thing Rufus doesn't detect well is if you're going to have like an expansion or contraction of microsatellite region. So if you go from 50 AT to 51, since that's longer than your Kamer size, there's actually no different Kamer. But FreeBayes will find that. So that's like the one case where mapping is going to find variant.
So microsatellite variants are difficult if they're longer than the Kamer size, right? Because whatever you see in the 25 MERS, there's no difference. You really have to look at the whole read to find the difference.
A variant, like an AT gets a G in the middle, will find that.
Yeah.
But it won't find that expansion or contraction.
Yeah. Instead of 58, you have 60 copies of ATs. Then any Kamer you examine is actually shared between the two sequences. Yeah. 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 So what you guys are going to be doing after this, I assume there will be a little break. And then after that, L's going to come on the scene, and you guys are going to be doing exercises for variant identification. He created some data sets, and then so you're going to go to SNP calling primarily, or short variant calling primarily, right?
Yes,
mapping. Yeah. And then he's gonna show some of another direction that we're going in the lab, and that's these interactive tools where you can actually examine the sequences and variants in basically in real time. So that will be on the menu until a little after lunch, right? Till 3. Okay. Well, if there's no more questions, then thank you for your attention.